Howdy friendos, Basker here. I really didn't mean to work on this video for so long, but me being the genius that I am, thought that it would be easy to write, record, and edit a video on a new virus causing a global pandemic, while also working full-time, getting school stuff settled, and writing scholarship essays. If this doesn't show my genius, I don't know what will. Our understanding of how SARS-CoV-2 infects cells and causes COVID-19 has been greatly expanded over this last year. Despite this, there are still several nuances to every infection which varies between individuals. Because of this, it's easier to do a kind of general overview of the infection cycle with the major types of cells and their roles in the immune response, as well as how they're affected. In order to cover as much as possible without getting lost in the forest of terminology, I'll be looking at two types of infections, asymptomatic and symptomatic. I also want to be clear on what I'll be detailing in this video. I'll be reviewing what's occurring within the body and some of the symptoms that come with it, but not all of the symptoms that come with the actual infection. The reason being that some of the symptoms lack a clear cause. I hope you like cytology and histology because this video is going to be all cytology and histology all the time. Before I start talking about the difference between asymptomatic patients and symptomatic patients, I want to correct some things that I had stated in my first video. I had stated that SARS-CoV-2 had evolved to its current state roughly 40 to 70 years ago, but what I should have said was that there was a speciation event where RATG13 and SARS-CoV-2 became two separate species. I also erroneously said that TMPRSS2 is what cleaved the furin cleavage site. In reality, SARS-CoV-2 can infect cells through either direct membrane fusion or endosomal membrane fusion. Direct membrane fusion occurs at the cell surface where the two membranes fuse and the viral genome is released directly into the cytosol, which occurs when the spike protein attaches to ACE2, then becomes cleaved by furin, a protein that is used to essentially remove a part of the molecule in order to expose portions that allow bonding to take place, which is along the area between S1 and S2. Once S1 and S2 are cleaved, the S2' cleave site is exposed. The newly exposed site is where TMPRSS2 cleaves the area again and allows the two membranes to fuse directly. Endosomal membrane fusion, on the other hand, requires that SARS-CoV-2 bind to ACE2 before being brought into the cell through endocytosis. Endocytosis is used by cells to bring in large particles and allow for better regulation of what comes in and out. Once attached, the virus is steadily drawn in through vagination before being pinched off and allowed into the cell within an endosome. The endosome eventually cleaves the virus through either cathepsin B or cathepsin L, both of which are proteins that degrade other proteins when endosomes are fused with lysosomes, which results in the fusion of the endosome and viral membrane. The mix-up of furin and TMPRSS2 came from my attempt to somewhat simplify the process, as my goal with these videos is to provide enough information that a layperson can pick up details and get a better understanding about what's occurring. This sometimes requires skimming over the finer details for a broader picture, and sometimes the information gets mixed up. The first type of infection I want to start off with is asymptomatic. Asymptomatic simply means that the infected person lacks any obvious symptoms, although this doesn't mean the virus isn't replicating. Why one person is asymptomatic while another becomes symptomatic isn't all that clear currently, but the most prevalent hypothesis is a faster and weaker immune response by the asymptomatic person, shown by the different expression of cytokines, which are small proteins used for cellular communication. An asymptomatic person will likely have lower amounts of tumor-necrosis-factor related apoptosis inducing ligand, TRAIL, macrophage colony-stimulating factor, M-CSF, growth-regulated oncogene alpha, GRO-alpha, interleukin-6, IL-6, granulocyte colony-stimulating factor, GC-SF, interleukin-2, IL-2, and tumor-necrosis-factor alpha, TNF-alpha. These lowered levels of cytokines point towards a more subdued immune response where the removal of infection takes place in the proverbial background, while other cytokines such as stem cell factor CF, interleukin-13, IL-13, leukemia inhibitory factor LIF, and interleukin-12p40 IL-12p40 tend to be much higher in the asymptomatic person. The cytokines listed above play roles in reducing the inflammatory response, such as LIF changing the pro-inflammatory response to an anti-inflammatory response, and IL-12p40 helping cells change so that they can produce the anti-inflammatory response. Some non-immune cells will also produce antimicrobial compounds when activated by IL-13. namely an increase in mucin 5AC. There is also some evidence that suggests that those whom came into contact with other coronaviruses, such as HCoV-OC43, HCoV-229E, HCoV-NL63, and HCoV-HKU1, are more likely to become asymptomatic compared to those whom haven't. I know that's a lot, but it's important to separate what may cause someone to be asymptomatic versus symptomatic. I'll also provide the context for each cytokine when it becomes important and how they interact with cells to create the immune response in the symptomatic case. I'm also going to go through most of the cells that become infected, but not all of them. The cells that I am going over are the ones that I found to be the most influenced by the virus. And like my last video, I'm going to include all acronyms in a list within the description. For the sake of organization, I'm going to start off with the upper respiratory tract. Most SARS-CoV-2 infections occur through the inhalation of respiratory droplets, with a relatively small amount being from viral covered surfaces. That's not to say that you can't be infected by touching that surface and then touching your face, but that you're more likely to get infected from aerosols released by the person than the surface that they spat on. Remember to wash your hands thoroughly, kiddos. When talking about the process of COVID-19, there's 3 phases of the disease: early infection phase, pulmonary phase, and hyperinflammation phase. The early infection phase is the one associated with the symptoms most people think of with the mild form of the infection, such as a dry cough and a fever. So once inhaled through the nose, SARS-CoV-2 immediately goes to work by infecting the cells found in the nasal epithelium, such as goblet cells, ciliary epithelial cells, and basal cells. Goblet cells are the cells which produce mucus, which is a combination of proteins called mucin 5AC and mucin 5B, a variety of salts and water. Ciliary epithelial cells are those that reside around the goblet cells and help get rid of any irritants in the mucus by beating their cilia, which are hair-like structures. That causes the irritants to move further up to the surface where it can be expelled. Basal cells are progenitor cells that are able to differentiate into other cells found in the epithelium and act as a backup to replace any damaged or killed cells. There are a few hypotheses on how the virus is able to permeate mucus, one being the viral particle hitching a ride on a bacteria that's able to essentially swim in the mucus. Once SARS-CoV-2 is able to move through the mucus, it attaches to the ACE2 receptor-rich epithelial cells and begins a replication process. When SARS-CoV-2 reaches the olfactory epithelium, the part of the nose responsible for the sensation of smell, it starts to destroy the cells that help support the olfactory sensory neurons. The damage becomes severe enough that the person loses their sense of smell and taste. There currently isn't a lot of evidence that supports the idea that the virus can infect neurons, but I'm keeping an eye out for any. In regards to the immune response, I'm saving that for when the virus enters into the lungs, as that's where the deadlier part of the infection takes place. As the epithelial cells become infected and begin to die within the nose, airway, and bronchioles, they commit a type of self-destruction called pyroptosis that starts the inflammation process. An inflammasome is formed within the infected cell due to the activation of a receptor that lines the inside, called retinoic acid-inducible gene 1-like receptors, or the way easier RIG1-like receptor, that is activated upon the detection of a virus's genetic material. The activation triggers the creation of a couple of interleukins, specifically interleukin 1 beta, IL-1 beta, interleukin 18, IL-18, as well as the creation of the protein Gasdermin D, or GSDMD. The interleukins are used to initiate the start of inflammation by acting as distress signals to other epithelial cells and immune cells, while the GSDMD acts as a way to cleave the cell and allows the interleukins into the extracellular space. Basal cells and club cells and begin differentiation into goblet cells, which in turn begin producing mucus. Normally this would be a good thing, as it would replace those goblet cells that had died, but doing so causes an excess of mucus, which in turn will cause a runny nose, and when draining into the back of the throat, will end up causing a dry throat as well. Once the virus passes through the bronchioles, they move into the alveoli that's composed of 3 different types of cells, all of which have the most inventive names ever: alveolar type 1 cells, alveolar type 2 cells, and alveolar macrophages. Here's where I'm going to start detailing the immune response, so strap in. First though, I need to say what each cell is responsible for. Alveolar type 1 cells are elongated cells that complete the gas exchange of oxygen and carbon dioxide, while alveolar type 2 cells have much more going on, as they secrete antioxidants, anti-inflammatory, and antimicrobial substances, as well as a glycoprotein called surfactant. Surfactant helps keep the alveoli dry, stabilizes the shape and size of the alveoli, and aids in maintenance of lung volume during respiration. Type 2 cells also act as progenitor cells for type 1 cells, both of which have a high quantity of ACE2 and TMPRSS2 receptors. Since both types are epithelial cells, they undergo pyroptosis as well. Within the alveoli, alveolar macrophages do their best to phagocytose viruses, loose viral RNA, and bits of dead cell floating through the surfactant, while dendritic cells within the lamina propria, which is the area just below the epithelial cells, pulls bits of the virus from the alveolar space above by parts of their membrane squeezing between the epithelial cells themselves. The antigens are then placed onto the cell surface through the major histocompatibility complex, MHC, which will become important later. Mast cells, also found in the lamina propria, react to the multiple cytokines and begin to release histamine. Histamine binds to the H1 receptor on endothelial cells that line blood vessels, and causes vasodilation, which is whenever the smooth muscle cells relax. This in turn causes the alveoli to flood with interstitial fluid. The epithelial cells also release M-CSF, which signals more macrophages to the area, as well as promotes macrophage creation from the bone marrow, and grow alpha, which recruits neutrophils to the area. Macrophages will also release GM-CSF, which serves to increase the production of granulocytes, such as mast cells and neutrophils. TNF-alpha is a cytokine that mediates the release of IL-1 and other macrophages as well as recruitment of more neutrophils, and IL-6 is a pro-inflammatory cytokine that becomes more important later. Neutrophils are similar to the alveolar macrophage as they phagocytose viral particles and other foreign bodies, but lack the antigen-presenting capability of the macrophage. Neutrophils also commit apoptosis instead of pyroptosis upon consuming a pathogen. Within the interstitial fluid also comes natural killer cells. Natural killer cells, NK cells, are responsible for killing infected cells. MHC1 returns when it comes to the interaction between the infected and NK cells, as infected cells downregulate their expression of MHC1, which the NK cells picks up on and immediately begins releasing perforin and granzymes into the targeted cell that causes the cell to essentially rip open and prevent the virus from replicating. The mixture of interstitial fluid, dead cells, and cell parts accumulate, which results in the formation of pus within the alveoli that inhibits the exchange of carbon dioxide and oxygen. This leads to a worse cough within the person as their body attempts to clear out the pus, as well as shallow breathing, as their bronchioles leading to the alveoli begin to constrict. This is called pneumonia. IL-1 beta and IL-6 are also responsible for starting a fever, but I'm not going to go into too much detail since those infected tend to die from other complications besides fever. Once pneumonia sets in, the second phase of COVID-19 begins. The pulmonary phase can be broken into two possibilities: pneumonia with hypoxia or without hypoxia. Hypoxia is a condition where oxygen can't reach specific areas of the body, which obviously is not a positive. Those that develop hypoxia normally require hospitalization and oxygen. The second phase is relatively simple to explain as all the cells involved are the same as before. The third phase, the hyperinflammatory phase, is considered the most lethal. This phase sees the introduction of the adaptive immune system. Eventually, the dendritic cells move through the pulmonary lymphatic vessel into the lymph node. Once in the lymph node, the dendritic cells activates both CD4 T cells and CD8 T cells with bits of the virus. The CD4 T cells then proceed to activate B cells, which in turn differentiate into plasma cells and memory B cells, as well as release IL-2. Memory B cells are important for long-term immunity, but don't play a large role in what occurs during the primary infection. The plasma B cells proliferate rapidly and are responsible for the release of immunoglobulins, specifically immunoglobulin M and immunoglobulin G, or IgM and IgG respectively. In the start of the adaptive immune response, IgM is produced first by short-lived plasma B cells in order to help fight the infection before the longer-living plasma B cells differentiate and start to produce IgG. IgG is much smaller than IgM, which allows IgG to move into the tissue of the infected site and bind to free-floating viral particles. These bound viral particles are unable to further infect cells and allows them to be phagocytized easier by immune cells. Now back to the CD8 cells, they act in a similar fashion to NK cells, but do so specifically through MHC1 antigen presentation instead of the multiple receptors available to the NK cells. Massive introduction of even more immune cells combined with their release of cytokines leads to further vasoconstriction and inflammation within the lungs and bronchioles. IL-6 plays a key role in the lethal aspects of COVID-19, such as hypercoagulability, acute respiratory distress syndrome, ARDS, and multiple organ failure. In order to get the full picture though, I need to go back through the system that's used by the ACE2 receptor, the angiotensin-renin-aldosterone system, also called RAAS. As I said in the other video, this system helps regulate blood pressure, with ACE2 specifically lowering blood pressure by converting angiotensin 2 to angiotensin 1-7. Angiotensin 1-7 acts as a negative feedback loop to angiotensin 2 by decreasing blood pressure through vasodilation. Reduction of ACE2, brought about by downregulation from infected cells, causes an increase in angiotensin 2, ANG2, that acts as both a vasoconstrictor and a pro-inflammatory cytokine, as well as an activator of ANG2 type 1 receptor AT1R, which in turn causes a positive feedback loop. The positive feedback loop leads to the massive increase of IL-6 that in turn causes more inflammation within the lungs. The inflammation continues until ARDS occurs within the infected person. IL-6 also travels through the bloodstream and causes inflammation throughout the body through similar receptors of the epithelial cells within the lungs, especially those within capillaries, which aren't much used when constricted. Release of the virus into the bloodstream is the current hypothesis for how it manages to reach the gastrointestinal tract. The virus continues to cause damage everywhere it goes and elicits the body's natural healing response, namely the accumulation of platelets to the area. Clots rapidly accumulate within various organs, spurred on by their activation by IL-6 until they create blood clots. These blood clots end up depriving cells beneath the clot of oxygen, which ends up leading to necrosis, and in turn causes the organ's loss of function, leading to multi-system organ failure and, obviously, death. Those that don't die of COVID-19, whether they live through an acute or severe case, typically have lingering effects caused by the disease. How long those effects remain are currently unknown, but it most likely depends on which organs were affected and how badly they were affected. In regards to the heart, it's also overly abundant with ACE2 receptors, but unfortunately there's not enough conclusive evidence to show whether damage to the heart is caused by the virus itself or the immune response to the virus. This is also why I didn't want to go into too much detail on brain fog, as it's not known if it's direct damage to the brain or if it's caused by hypoxia. When it comes to the effect on kids, there's also not a large amount of information on why children seem to be much less affected than adults. Despite this, they can still act in a similar manner as asymptomatic persons, as viral loads between the two are roughly the same. Whew, okay, well that was a lot. Trust me, there's a lot more to the infection cycle, but this is just a general overview that gives a broad explanation of the symptoms and the causes. I encourage everyone to read through all of my sources and double check what I've said in this video. Let me know if I can do better on any explanation or if there's something that I need corrected. I want to avoid putting out any misinformation accidentally, and the only way that I can do that is to be actively fact-checked. And just always remember, friendos, challenge your knowledge.